Hello, everyone, and thank you for joining us today. My name is Felicia Goodrum, and I am the co-editor-in-chief of the Journal of Virology, or JVI. The American Society for Microbiology, or ASM, publishes JVI, which is hosting today's webinar, as well as the others that we call the JVI Seminar Series. For more information on the seminar series and to watch our introductory episode, please visit the JVI website or scan the QR codes you see on the slide here. If you are not familiar with JVI, we publish the preeminent work in virology research. We welcome primary research, mini reviews, and other article formats that investigate fundamental processes and viral structure and mechanisms by which viruses interact and evolve with their hosts and environment, as well as novel methodologies, therapeutic, and diagnostic strategies. Before I turn things over to our speakers, let me just quickly go through the format of our webinar. The first 35 minutes will be about the science and the presentation by our guests. We'll then have about 10 minutes for questions and answers, and the remaining 15 minutes will be devoted to the discussion of the manuscript and how we need to advocate for science and its importance to public health. We'll answer such questions as why the study is important, what does the study mean for your family, neighbors, or the public, and we'll also ask the speakers to share their experience in going into science and what they would like you to know about their career as a scientist. So a few housekeeping rules. This webinar and future JVI Seminar Series webinars will be recorded and posted to the JVI website so that you can always access them. Our plan is to host these webinars every other month. And in lieu of a certificate of attendance for these, you will receive an email confirmation for registering for the webinar, and this serves as proof of your attendance. We do not provide certificates of attendance. If you have any questions for our speakers, please use the Q&A button at the bottom of your Zoom screen. We are not using the chat function, so please submit all questions through the Q&A. Our webinar today is entitled Uncovering Cryptic Genes, Functional Dissection of HSV-1 UL 31.6 as a neuron-specific virulence factor. Our speakers will discuss their paper that was an editor's pick and was published in JVI in July of 2024. If you've not read the paper, I highly encourage you to do so. It's free and accessible from the JVI website. Okay, now I will invite Dr. Anna Cliff, one of JVI's editors, to introduce our topic and speakers today. Anna?
Thank you, Felicia. So I am Anna Cliff. I'm an associate professor at the University of Virginia and also one of the editors at Journal of Virology. So I'd like to introduce our speakers. So we have Akihisa Kato and Yasushi Kawaguchi from the Institute of Medical Sciences in Tokyo, Japan. And as Felicia mentioned, this article was published last year, and I was the handling editor, and I picked it for this JVI seminar series. I picked it for many reasons, but I think mainly because it highlights just how much we still have to learn, especially about herpes viruses and the function of the genes that they encode. So these are highly evolved viruses, right? And one reason that they are, so successful is that they have to pack so much genetic information into their genomes. And yet we're still learning more. And there have been numerous kind of multi-omic studies over the past few years that have just, uncovered potentially novel open reading frames. So this study builds on this by showing that there's a novel open reading frame that encodes protein, and they use really beautiful then classical virology to show that this protein has a role in viral pathogenesis. And I don't want to take too much away from the speakers now, so I'm going to hand over to them and they're going to introduce themselves and take us through their study. Thank you.
Okay, can we start? Okay, can I share the slide? Okay, can you see the slide? Okay, thank you for the introduction. My name is Yasushi Kawaguchi. I'm professor and PI in the Division of Molecular Biology of the Institute of Medical Science, the University of Tokyo. Before the presentation by Akihisa, who is the first author of the paper, I would like to briefly introduce the virus which we focus on today and overall research of our laboratory. As shown in the slide, the virus which we focus on today is herpes simplex virus, HSV, a prototype of herpes viruses. This virus causes a variety of diseases in humans, including life-threatening encephalitis, eye and skin disease, and genital herpes. Actually, I began my research on human herpes viruses in 1995 at the University of Chicago in the laboratory of Professor Bernard Roisman. Since my first publication in 1997 on the interaction of HSV regulatory protein ICP0 and host factors, I have published 72 papers in the Journal of Virology and MBio, as well as well-established journals listed here as either the first or corresponding author. Our research is characterized by investigating a wide variety of aspects of virology with the aim achieving a comprehensive understanding of the molecular mechanisms underlying the replication and pathogenesis of HSV. Okay, now I would like to hand over to Akihisa. Akihisa, please.
I'm Akihisa Kato, an associated professor in Professor Yasushi Kawaguchi's lab. It's a great honor to have the opportunity to present our research at the JV webinar. I would like to thank the organizers for this opportunity. Today, I will introduce our in-depth analysis of the HSV-1 cryptic orphan gene UL31.6 that we recently identified. Professor Bernard Roisman's review in 2030 mentioned the genome of HSV contains at least 84 different coding sequence CDS. Recent multi-omics analysis has revealed HSV-1 additionally encoded more than 200 potential cryptic orphan CDS. Among these multi-omics approaches, we developed a unique system to identify cryptic orphan HSV CDS at peptide level. Many viruses, including HSV-1, are known to shut off host cellular protein synthesis. In other words, the majority of the newly synthesized protein in infected cells are thought to be viral proteins. This unique feature of viruses led us to hypothesize coupling selective enrichment of newly synthesized protein with high-resolution mass spectrometry analysis might allow us to identify previously unreported viral CDS. To test this hypothesis, we performed chemical proteomics integrating Bonk technology and high-resolution mass spectrometry analysis. As you know, protein translation begins with methionine. Therefore, in this study, newly synthesized proteins in HSV-1 infected cells were labeled with a methionine analog, AHA, in methionine-free medium. AHA-labeled proteins were then purified using alkene-conjugated agarose by quick chemistry, digested with trypsin, and subjected to high-resolution mass spectrometry analysis. By using this system, we identify 9 cryptic orphan CDS, IUL12, IUL26A and B, IUL38, IUL44B, IUL48, 49, IUL50, and IUS1, whose translated products are expressed in HSV-1 infected cells. Furthermore, we showed a newly identified HSV-1 protein, PIUL49, encoded by IUL49 gene, is critical for neurovirulence in mice by regulating viral DUTPS, a key enzyme for HSV-1 genome integrity. As I mentioned earlier, recent multi-omics analysis have revealed HSV-1 additionally encodes more than 200 potential cryptic orphan CDF. However, only a limited number of the cryptic orphan CDF has been verified to be expressed in HSV-1 infected cells at the peptide or protein level. And among them, PIUL49 is the only viral protein whose biological significance during HSV-1 infection has been reported. Considering these findings, we believe a better understanding of viral pathogenesis requires not only the identification of such cryptic orphan CDS, but also their in-depth characterization. In our previous study, 9 HSV-1 CDS were identified based on the following relatively strict criteria. Criteria 1, at least 2 distinct peptides detected in HSV-1F infected cells, each of which was identified by more than 1 peptide spectrum match, PSM, were mapped to a previously unannotated CDS. Criteria 2, the sequence of CDS was conserved in another HSV-1 strain, HSV-170. In the present study, we reanalyze the chemical proteomics data based on the following alternative criteria. In particular, criteria 1 was modified. Instead of requiring 2 peptides, it's supported by more than 1 PSM. We now require at least 3 distinct peptides is supported by at least one PSM. Criteria 2 remains unchanged. The analysis of the previous chemical proteomics data identify additional two cryptic orphan CDS, UL 9.5 and UL 31.6. In this study, we focused on UL 31.6, which was previously identified by ribosome profiling by Dolgen's group and by our chemical proteomics. As shown here, chemical proteomics of HSV-1 infected cells detected 6 different peptides derived from PUL31.6. To verify, these peptides correspond to the full-length PUL31.6 expressed in HSV-1 infected cells. We generated a recombinant virus, UL31.6, predicted to express PUL31.6 tagged with a monometric fluorescence protein, Venus. As you can see here, anti-GFP antibody, which also detects Venus, recognized an approximately molecular mass of 56 kilodalton from lysate of cell infected with Venus UL31.6, but not from lysates of cells mock infected or infected with wild-type HSV-1F. We also generated a specific antibody against PUL31.6. As expected, the antibody against PUL31.6 specifically reacted with a protein with approximately 37 kilodalton in lysates of cell infected with wild-type HSV-1F, but not from lysates of mock infected cells. And the mutation in start codon of UL31.6, which was designed to have no effect on amino acid sequence overlapping UL32, abolished the about 37 kilodalton band in HSV-1 infected bear cells, but had no effect on the expression of UL32. And importantly, this about 13.7 kilodalton band was conserved across different HSV-1 strains, including HSV-1, VR3, and HSV-1-17. These results indicate PUL31.6 is expressed in HSV-1 infected cells as a protein of approximately 37 kilodalton. HSV-1 genes are expressed in a cascade classified as immediate early alpha, early beta, leaky late gamma 1, and true late gamma 2. As reported previously, inhibition of viral DNA synthesis by PAA reduced the expression of gamma 1 protein, VP16, and severely impaired the expression of gamma 2 protein, US11. In the presence of PAA, the accumulation of PUL31.6 was significantly reduced, but remained consistently detectable, as observed with that of VB16, a gamma-1 protein. These results suggest UL31.6 is a gamma-1 gene, and it encodes a gamma-1 protein in HSV-1 infected cells. As you can see here, PUL31.6 was also detected in virions purified from HSV-1F infected Vera cells, suggesting PUL31.6 is incorporated as a virion protein of HSV-1. Where is UL31.6 located? Within the three compartments of virion: envelope, tegment, or capsid. To address this question, we performed tegment release assay, reported by Morrison's group. In this assay, purified virions were treated with detergent to remove the envelope proteins, and tegment proteins were dissociated from the virions in high salt solution. The sample were then separated into supernatant and pellet fraction by centrifugation through a sucrose cushion. In the absence of high salt, PUL31.6 and major tegment protein UL47 were detected in the pellet fraction. In contrast, the envelopes protein GB was detected in the supernatant fraction. In the presence of salt, the detection of PUL31.6 and UL47 shifted from pellet fractions to mainly supernatant fractions. Meanwhile, capsid protein VP5 still detected in the pellet fraction. These results indicate PUL31.6 is a tegment protein. The next question which we addressed was the biological significance of PUL31.6 in HSV-1 infection. The UL31.6 null mutation had minimal effects on viral growth kinetics, plaque size, and the accumulation of HSV-1 proteins expressed from genes of all kinetics class alpha, beta, gamma-1, and gamma-2 in Vera cells. Furthermore, virion morphogenesis in Vera cells infected with UL31.6 null mutant virus was similar to that in cell infected with wild-type HSV-1F. Notably, the overlapping UL32 is known to be critical for capsid maturation in infected cells, indicating the UL31.6 null mutation employed in this study had no effect on the function of overlapping UL32. These results indicate the UL31.6 plays no obvious role during HSV-1 infection in Vera cells. How about in vivo? To investigate the effect of PUL31.6 in vivo, we first employed a murine model of herpes simplex encephalitis induced by intracranial infection. This survival curve shows the UL31.6 null mutation significantly reduced the mortality of mice following intracranial infection. In addition, this mutation also significantly reduced viral replication in the brains of mice following intracranial infection, indicating PUL31.6 is a novel neurovirulence factor of HSV-1. We next examined the effect of PUL31.6 in a murine model of herpetic keratitis and skin disease induced by ocular infection. In contrast to intracranial infection, the UL31.6 null mutation had minimal effect on the development of HSK and periocular skin disease in mice following ocular infection. Similarly, this mutation has minimal effect on viral titers in the tear films of mice following ocular infection, indicating PUL31.6 is required for efficient HSV-1 neurovirulence, but not for ocular infection in mice. Next, we investigated whether its function might differ depending on the type of cultured cells. To investigate cell type dependent effect of PUL31.6 in cell cultures, we determined plaque sites of wild type HSV-1F, UL31.6 null mutant virus, and its repair virus in several cell lines, including human neuroblastoma, SKNSH, and CHP134 cells, and mouse neuroblastoma, neuro2A cells, human carcinoma, HEP2 and A549 cells, and mouse fibroblastoma, NIH3T3 cells. As you can see here, the UL31.6 null mutation significantly reduced plaque sites in all human and mouse neural cells tested. On the other hand, this mutation had minimal effect on plaque size in all human and mouse non-neural cells tested. These results indicate PUL31.6 is required for efficient HSV-1 cell-to-cell spread in a manner dependent on cell type. Having established the functional role of PUL31.6, we next examined its evolutionary conservation among herpes virus. Bioinformatic analysis showed the CDS of UL31.6 is conserved among the sequence all HSV-1 strains in the NCBI database. And UL31.6 homologs seem to be shared only in a few members of genus simplex virus in subfamily alpha herpesvirinae, including herpes simplex virus type 2, HSV-2, chimpanzee herpes virus, CHHV, and spider monkey herpes virus, HVA1. Let me conclude our presentation. What we found is PUL31.6 is a tegment protein, promotes viral cell-to-cell spread, specifically in neural cell culture. Is required for efficient viral replication and pathogenicity in the brain's mice, and is conserved among all HSV-1 strains analyzed and in certain members of the genus simplex virus. Taken together, our series of observations indicate a tegment protein PUL31.6 act as specific neurovirulence factor in HSV-1, potentially by promoting cell-to-cell viral spread in neural cells. Finally, I would like to acknowledge Toru Natsume, Shungo Adachi, Hideo Kusano for mass spectrometry analysis, Shoichi Kawano for bioinformatics analysis, all members of Kawaguchi Lab, and my mentor, Yasushi Kawaguchi, shown on this slide. That's it from me. Thank you so much for listening.
It's wonderful. Thank you so much. So we have questions that have come in both on the chat, and I have some that were sent in prior to the seminar. So I'm going to start with some that came in through the chat. So the first one is from Yun Sok Cho. It says, why did you change the reanalysis criteria?
Okay,
Why?
Okay, I would like to answer. Okay, the initial criteria was very strict. One, point out by a reviewer during the review of the first paper in Nature Communication. And we knew that under such a strict criteria, some unknown CDS were excluded. This is the reason. Okay.
And then Orkidi Koyunchi has a question, which is, what is the proposed mechanism of neurovirulence of this novel gene? And is there a difference in replication efficiency of the null virus mutant in peripheral versus central nervous system neurons?
Akihisa?
Precise mechanism is unknown, but in our study, the expression of PUL31.6 is higher in neural cell line than other cell line. So I think high expression of UL31.6 is a very important clue.
Can I ask you just a follow-up question as well? Because I just think this is super intriguing. Did you look, or have you done follow-up studies to look then from when you do the eye infections into the central nervous system? And is there a change? Because, you know, if it was blocked in trafficking to the CNS from the periphery, I think be a super cool target.
We didn't perform such experiment. But it's very interesting.
Yeah.
So in future we will perform such experiment. So in response to the previous questions, so it's very difficult to unveil the mechanism by which this new neurovirulence factor act in central nervous system. Probably we will perform the interactome to identify the host factor which interact with this neurovirulence factor to obtain the first clue.
Yeah, super interesting. Okay, so from Elena Kechititin, I apologize if I just butchered that, I'm sorry. CMV and some other herpes viruses don't have UL31.6, but still infect the CNS, especially in immunocompromised patients. Do they express an equivalent protein like VZV does, or do they use different mechanisms to invade the CNS? I guess my first question as well, following up. So does VZV have a homologue? Have you looked?
Akihisa?
No. BZB did not the homologue of this gene.
Mm-hmm.
Actually, okay, as a many alpha herpes virus, such as Sugar rabies virus, do not have this homologue. Okay, only HSV-2 chimpanzee herpes virus and
spider monkey.
Yeah.
And spider monkey herpes virus, just four virus.
Yeah. Mm-hmm. Encode this virus, this protein. It's very limited.
Okay, and I have a question that was submitted. Oh, actually, sorry, I'm missing some in the chat. So Roger Lepay says, very interesting talk. Can your approach detect alternative start sites? For example, the second methionine residue as for GM or other amino acid?
You mean Boncat system? No. You mean chemical proteomics capture non-AUG ORF? No.
That unless Roger wants to type more.
Sorry.
Okay, actually, okay, I will answer. Okay, so I would not. We didn't do that. Okay, so we don't know at present whether the second there are alternative site in this CDS. We don't know, but it's very interesting question. Thanks,
So,
Roger.
were you able to detect UL12.5, which would be, I guess, an alternative start site for in UL12 as well?
Pardon me?
We can move on. I was just trying to help determine the answer. Okay, so let's see. We have a question that says, which host cell receptors or proteins does UL31.6 interact with to mediate its neurovirulence? I think you've kind of answered that by saying you don't know, right, the mechanism. I mean, you're doing the proteomics. David Levy asked, does loss of 31.6 have an impact on viral latency in neuronal cells?
I don't know, but yeah, very interesting question.
Yeah, if so, it's wonderful.
Would like to, yeah. In the future, I would like to investigate this point.
Okay. So Roger does have a follow-up here. Another way to ask is if the modified methionine is only added as first met or for all met residues a longer protein.
Wow. So the ask is if the movement is only
Maybe we can
Akihisa?
answer that one offline later.
Hmm?
I have a couple as
Yeah.
well, I think, that were submitted beforehand. So I guess, maybe we don't know this, but are there any specific tegument interacting proteins identified for UL 31.6? What might that apply for virion assembly?
Interaction. Sorry, I can't catch the question. So
Did you not catch it? Just if there are any tegument interacting proteins identified, what it might be.
okay. I don't know. We don't have any data, but I think UL 31.6 interact as a tegument protein. This is important question.
Okay. We'll
Okay.
come back to. Did you
I would
see
like
Adam
to-
Wisnett?
Okay, I would like to respond to Roger's questions. So the all methionine are labeled, not only for the initial methionine. Okay.
There was a follow-up question as well to Rogers from Adam Wisnett, who says, following on Rogers' question, have you looked at which non-canonical start sites CUG are missing from your data set but show up in the mass spec? There's some reports leucine can initiate, but I've never seen a clickable leucine. Everyone just kind of assumes M is the start.
Which one?
Actually, maybe actually AHA is analog of methionine, but many proteins have not only methionine. So our system detected non-AUG protein, and we used a comprehensive database for mass spec analysis. So our database cover non-AUG proteins. So maybe our the database of chemical proteomics cover non-AUG protein. But we search only first codon is AUG CDS. So we may not catch the type CDS. Is okay? Oh.
So we only have a couple of minutes left and we have a very mixed audience here. So I'm going to pick another question from those submitted beforehand that's just a little more kind of open and general. And that is, what motivated you to focus on this specific virus host interactions, and what were the findings that most surprised you?
Yeah. But now we don't have such data, but this is important point. So we will investigate in future.
Yeah, I mean, I think to me the most striking is just the difference in the spread in the different cell types, that it was this new, of course, like the complexity of the genome and being able to identify this novel peptide, but then that it's required for neuronal spread, I think is super interesting.
It's so interesting that these herpes viruses are so complex. They infect different cell types and they have to have different genes that function in different places. I agree with Anna.
Yeah. Okay, well, wonderful. I think now I'm going to turn it back over to you guys and Felicia for the next part of the webinar. Thank you so much.
You.
Thank you for a wonderful presentation. So science is a systematic and evidence-based pursuit of knowledge through rigorous and controlled observation and experimentation. It's really our best hope for addressing the challenges to human, animal, and plant health, and for the betterment of the quality of life. It informs decision-making like nothing else can. So herpes simplex 1 or HSV-1 is one of 9 herpes viruses that infect humans. Its infection presents most typically as cold sores in the oral region or fever blisters, and it's spread by direct contact. The majority of the population worldwide is infected with HSV-1, whether or not they present with cold sores. Like all herpes viruses, HSV-1 will establish latency, and this allows continual reoccurrence of disease over a lifetime. Herpes is forever. So while cold sores can be really bothersome, HSV-1 infection is also the cause of more dangerous and rare, thankfully, diseases such as encephalitis, and it's really dangerous in newborns. So I really want to thank you both for your work and your presentation. And I'd like to talk a little bit about why your results matter. Why is it important that in 2024, you have identified a new herpes virus 1 protein?
Akihisa?
I think this study is important for two main regions. In basic virology, we believe a deeper understanding of viral pathogenesis requires not only identifying viral genes by omics analysis, but also characterizing their functions step by step. Since HSV-1 is a representative model of neurotropic viruses, identification its neurovirulence factor may provide insight for studies of other neurotropic pathogens. In applied research, we think decoding viral genes is essential because nearly all antiviral drugs and vaccine target viral proteins. And so moreover, identifying the new genes specifically contribute to neurovirulence of HSV-1 can directly enhance safety in the application of HSV-1 vector and to improvement of oncolytic virus for cancer therapy. This is my opinion.
Yes, I agree. It's really exciting after all these decades of study of herpes simplex 1 that we're still learning so much that's going to be important for controlling disease in individuals who are infected. I think that's really such a powerful thing that comes out of your science. So can you tell us a little bit, and maybe Yasushi, you could begin the answer to this question, but could you tell us a little bit about how you came to science and your career trajectory to approach these questions?
Okay. Okay. I'm getting old, so Akihisa is very young. So Akihisa will answer this question.
When I started doing research at the university, I found it's really exciting and enjoyable to uncover things that were not understood. This experience inspired me to go into science as a career. Of course, research can be challenging, but every new discovery makes it deeply satisfying. These days, here are younger people in Japan are going on to PhD programs, but discovery in basic research are very valuable, and the knowledge and experience you gain, so something special. You can also meet wonderful friends through science. I hope you will take on the challenge of science. Okay?
That is what we hope to help people do through
Yeah.
those webinars. So thank you both. I'd like to ask each of you to tell me a little bit about what you would tell those people who are watching that might be interested in entering a career of science. What should they know about this career? Akisha, do you want to start?
Right? Sorry, I can't get one more, please.
Sorry, I didn't understand.
Yeah.
Yasushi, what would you like to tell people? And we'll come back to Akisha. Yasushi, what would you like young budding scientists to know about a career in science?
Okay, say again.
What would you like young scientists or people who are interested in a career in science, what should they know about this career?
Oh, our career, you mean?
Career, the profession of being a scientist.
Korea.
Oh, okay, career, okay. Okay, so as Akisa mentioned, so doing science is very exciting because we can uncover unknown thing by ourselves. Okay, so that's an exciting thing. So that's why I choose science field for profession. But to continue profession in science is it's not so easy because we need to obtain good result, and we need to write a paper, and we need to obtain grant. But beyond these hard things, so to see the new things is very exciting for me. So that's why I'm doing science. Okay. Akisa?
Yeah, I think so. This is perfect answer.
I think one thing, Akisa, that you alluded to earlier is the global reach of science and making friends across the field. And I think it is wonderful the way science brings together scientists from all across the world, and that how your work on herpes simplex 1 may inform Anna's work on herpes simplex 1, and how through the papers that we publish, hopefully in Journal of Virology, that it really is something that allows science across the world to progress, and so that we have better understanding of the natural world and what causes disease and how we can end better lives. So I thank you both for joining us. Maybe now we will move to close our webinar and just give you a few reminders at the end that the Journal of Virology really wants your manuscripts. We encourage submissions covering all aspects of the field, and many of them are listed here. The Journal of Virology is the premier journal for virology research, and we are the journal that is working to support virologists in the field. And so we hope that you will continue to join our webinars, share them with your friends, and submit your manuscripts. And check out our seminar series. We have a new one coming up in November, and we'll be sending out notifications about the date of this seminar series and invitations for you to join us. And then you can also, on the Journal of Virology homepage, you can check out all of the seminars that have been previously recorded. And please contact us. We're really interested in your ideas for future webinars. If you read a fantastic paper in the Journal of Virology that you would like to see featured, please contact us. We'd be really interested in hosting papers that you have either published in the Journal of Virology or ones that just really excite you. So thank you for joining, and we look forward to seeing you next time.